Now, the brush border peptidases, as it says here, there's multiple peptidases present on the brush border of to the cytoplasm of the villus epithelial cells, and they have a different affinity for the relatively larger oligopeptides because they want to break them down. So there's three types of brush border peptidases. There's endopeptidases, there's exopeptidases, and there's dipeptidases. So going back for a second, the luminal protein digestion, the proteins are digested by endopeptidases to produce oligopeptides with 2 to 6 amino acids. And these oligopeptides, as a result of the protein digestion from the endopeptidases, they can either be large or they can be small. Now, first of all, let's have a look at what happens to the large oligopeptides. Well, the large oligopeptides need to be broken down even further, so they're digested by brush border peptidases to form amino acids. Whereas the small oligopeptides, they're transported into the enterocyte via pep-T1 cotransporter with hydrogen. So you've got the proton gradient here that helps basically energizes the pep-T1 absorption of small oligopeptides into the enterocyte. And once the small oligopeptide is inside the enterocyte, then it's digested further by cytosolic peptidases. Well, remember, it's either absorbed directly into the blood or it's digested by cytosolic peptidases. And that also provides the amino acids. And these amino acids, they're then transported across the basolateral membrane by various means into the blood. So we can summarize that here. Oligopeptides can be digested by brush border peptidases, like so, they're large, or they can be absorbed via pep-T1 and then digested by cytosolic peptidases, so the peptidases within the enterocyte cytoplasm. The uptake of the peptides, of the oligopeptides into the enterocyte, is by pep-T1, a proton gradient-driven cotransporter. So let's have a quick reminder of the routes of protein digestion. So the luminal proteases have broken down the proteins into oligopeptides. Some of the oligopeptides that were very small, they were taken up by pep-T1 into the enterocyte and digested by cytosolic enzymes to give us cytosolic amino acids. So the larger oligopeptides were digested by brush border dipeptidases, brush border peptidases to give us amino acids at the apical membrane, and some of the oligopeptides were further digested by exopeptidases to also give us amino acids at the apical membrane. So now we need to think, how are the amino acids imported across the apical membrane from the lumen into the enterocyte? And the way the amino acids are imported in is through four routes, basically, right? So these amino acids, you can categorize them into four categories essentially. The cationic amino acids, which are positive, and cysteine. The neutral amino acids, the anionic amino acids, so these are negative, and glycine and proline. So first, looking at the cationic amino acids and cysteine, how are they imported into the enterocyte from the lumen? Well, they're imported using a B0 plus cationic amino acid, neutral amino acid antiporter. So let me explain what that means. Well, basically, it's an antiporter, which means something goes in and something goes out, right? So what happens is that the cationic amino acids and cysteine, they are imported across the apical membrane into the enterocyte, whereas the neutral amino acids in the enterocyte, they're transported out into the lumen, right? So that's a B0 antiporter. And the important thing to remember is that this antiporter, it is sodium independent, right? And I'll explain why that's important in a second. The next category of amino acids is neutral amino acids, right? The neutral amino acids, they're transported in not using an antiporter, but using a B0 neutral amino acid sodium co-transporter. So what does that mean? Well, that means that basically the neutral amino acids at the apical membrane, they're transported across the apical membrane into the enterocyte along with sodium. So they require sodium for co-transport, and that's B0 co-transporter. And the reason it's 0 and not a plus is because it's not the cationic version, it's just the neutral version, right? So what does that result in? Well, it results in an import of amino acids and sodium across the apical membrane into the enterocyte. Now the next category of amino acid that we can look at is the anionic amino acids, and these are negative, and these are transported into the cell in a similar mechanism to the neutral amino acids. They're transported in via an EAAT3 anionic amino acid / sodium co-transporter. So what does that mean? It means that the anionic, the negative amino acids, they're transported across the apical membrane into the enterocyte along with 3 sodiums. So just like the B0 co-transporter, this is a EAAT3 co-transporter. And the way you can remember this one is EAAT is that it's anionic amino acids, the 2 A's, anionic amino acids, right? And again, that results in amino acid as well as sodium import, so it's sodium dependent, right? And finally, we have glycine and proline, which are 2 other amino acids in a set category on their own. Glycine and proline, they're imported across the apical membrane using this amino glycine proline sodium co-transporter. So, just like these 2, it's sodium dependent, it's a co-transporter, and it results in the import of sodium as well as the amino acids across the apical membrane. Whereas the B0 plus cationic amino acid / and cysteine neutral amino acid antiporter, that was sodium independent, and instead it required amino acids which are neutral being pumped back out into the lumen. So that results in the import of just amino acids. And that was the amino acids being imported across the apical membrane into the enterocyte. 2 more things we can note here is that, well, cystinuria, that's going to affect this B0 antiporter, and Hartnup disease is going to affect the B0 co-transporter. Now, once the amino acids have entered the enterocyte, 10% of them are used by the enterocyte for protein synthesis, right? So what happens to the rest of them? Well, the rest of them are, once they've been imported, they're then effluxed across the basolateral membrane into the blood. So this is how it happens. So the amino acids that are imported, they're put into 2 categories. They're either cationic, which are positive, or neutral. Now the cationic amino acids, which have been imported into the enterocyte, they're effluxed through a heteromeric 4F2HC lat antiporter. Whereas the neutral amino acids, they're effluxed through a 4F2HC independent. So this is 4F2HC, whereas this is 4F2HC independent lat transporter. So this is antiporter, this is a transporter. And what these two channels they do is that they transport the amino acids, they efflux it into the blood across the basolateral membrane. So that's amino acid efflux. So what have we done? So amino acids, the apical amino acids, they are imported and then effluxed. And 10% of them are used intracellularly for protein synthesis.